Welcome to another edition of the Wound Repair and Regeneration Fireside Chat. My name is Kyle Quinn, and I'm with my colleague Mitch Sanders, and we are co-chairs of the Wound Healing Society Communications Committee.
Wonderful. It is our pleasure to introduce Dr. Boris Hinz. Dr. Hinz is a distinguished professor of tissue repair and regeneration at University of Toronto, and he's one of 3 editors-in-chief of Wound Repair and Regeneration, along with Dr. Kirshner, Rob Kirshner, as well as Heather Powell. Boris, welcome. It's a real pleasure to have you here. It's exciting that we got to feature volume 29. I think it's issue 4 on Myofibroblasts. So welcome, and it's a pleasure to have you here.
Thanks, Mitchell. Thanks, Kyle. Well, you know better than I which issue it is. I just had to look it up just now.
It's not every other one. I think because I read that issue from cover to cover, and it was really outstanding. So anyway,
I'm impressed you have a print copy over there.
Yeah, I haven't seen one in a while.
So, Boris, tell us about this 50th anniversary that the Myofibroblast is having this year.
Right. That's an easy one. Usually say where to start, but that's easy to start because I should start in 1971, where the name Myofibroblast first came up. And well, I'm personally attached to that issue. Well, A, my research is on it, and B, I spent a postdoc period quite extensively in Geneva with Giulio Gabbiani, who was one of the discoverers of the myofibroblast. And that was done in 1971 in 2 papers, one in Science and one in Experientia, where they described fibroblastic cells or cells in granulation tissue in the rat, which had features of fibroblasts, endoplasmatic reticulum, and features of smooth muscle stress fibers. And looking back from where we stand now, we'd say, well, what's the big deal, right? What's special about that? The specialty was that at that time was still discussed how wound granulation tissue closes, so how wounds are closed, and reepithelialization was understood, and then the whole connective tissue part was not really clear. So people thought that it was a shrinkage of collagen still, which closes the wound. And together with other studies, more fundamental side, Giulio and the co-workers have shown that, well, there are actually cells in the granulation tissue, which at some point promote contraction from within. So it's like you're sitting in a sponge and you pull from within the sponge. So that's how the sponge goes down, rather than whatever squeezing out of water and collagen. And the other cool thing about this was also, along with a number of other studies in that field, is that cells which are not muscle cells or not smooth muscle or not skeletal muscle are able to promote to promote forces, to exert forces. So that was a novelty in finding these cells in physiological or pathophysiological conditions. That was a novelty which kind of founded a whole field these days. There's one other aspect. So the first work was was an ultrastructural description of those cells, and the function was kind of extrapolated. But then the follow-up study in Science was using strips of granulation tissue and doing all kinds of pharmacology on those strips that you usually would do on strips of vessels to see how they contract. And the authors found that a granulation tissue, given the right stimulus, can contract as the wall of a vessel, which I think I can't appreciate how cool that must have been at the time. Mm-hmm. So I did some of those studies. It's kind of impressive to see how a little piece of tissue, which is not meant to contract, actually does promote force.
So in your editorial, you talked about how the proposed origins of myofibroblasts have varied with different ideas proposed, discarded, and revived, and what makes the myofibroblast so enigmatic?
Yeah, if I only know. Yeah, so I worked on those cells for a while now too, and this is an issue I try to stay away from because people become very strong opinions about where these cells come from, and I understand it. So let's go a step back. so the description of myofibroblasts is a morphological one initially, or a functional one, meaning it's a contracting mesenchymal cell, so a cell which has not been contractile before. So if you look at a cross-section of our skin in connective tissue, you'd find cells with fibroblastic features which have no contractile elements, and that changes. So those cells acquire this phenotype. And originally, I think the fibroblast, whatever that is, was considered to be the precursors of myofibroblasts in all kinds of organs which have fibroblasts. And I think we actually already, not me personally, but people understood already in the 80s that there's a vast heterogeneity in the not only the precursors of the myofibroblast, but also in fibroblasts. And that has been revived now with much better technology. And the idea is, why would we care where they come from? It's because we have to target them, which I can talk a bit later. So what was the question again? No, the question is, why is it so enigmatic? Yes, why are we hitting each other about this question? Well, there's no good marker. That's it. So there is no good unique identifier to say, okay, well, I'll do a flow cytometry, I do a single cell RNA-seq. So this is the protein which comes up in these cells, and that must be a myofibroblast. That's just not how it works. And I believe the reason for that is because there's so many different progenitors, and the function of these cells is to quickly heal tissues. So jump in, produce collagen, fix whatever injury there is with super glue, and it has to be fast. And because the body or the organism can't really predict where an injury is happening, could be in the skin, could be in the heart, could be in the lung, there have to be local cells which are able to do that function, and they all converge to this very similar phenotype, meaning making collagen and pulling on that collagen. And they look alike. They're all different from where they come from. They're probably all different in their profile, but they're meant to do the same in this particular situation. And that's the problem. So if you want to have a marker, there's this protein called alpha smooth muscle actin, which is very frequently used. But the name already indicates a smooth muscle actin is also expressed in smooth muscle cells. But it comes up in activated versions of the myofibroblast at a very mature state of them.
I was really surprised based on RNA-seq that just fibroblasts can have at least 10 different lineages. And pretty remarkable.
Well, we'll see where this goes over the next 5 years. So how many more different lineages we find. It is true. In a way, it's surprising, but I think it's a okay, well, how do I phrase this without offending anyone? It's kind of a semantic issue in a sense. I know it's obviously a more profound problem, but the reason that we find 10 different or more different phenotypes or fibroblasts is just because we all grouped them before into one group because we didn't know better. We didn't have the tools to discriminate them. Now we have the tools, and I'm sure we can do a good job in coming up with 30 different subpopulations, and then we'll see where that goes and how that helps us.
It'll probably go the direction of the macrophage, right? Where eventually it'll just be like there's a spectrum of fibroblasts and myofibroblasts.
Yeah. And at some point, you're right? Then you call them M1s, M2s. Now we call them F1s, F2s. And then we'll figure out, okay, that's not really capturing it either. So we'll go back and say, they're plastic. They do things when they need to.
So you kind of touched on this, but many of us tend to think about myofibroblasts in the context of granulation tissue and its role in skin wound healing. But this issue highlights a number of other diseases where they play roles. Could you tell us about some of those applications?
Yeah, so I'm a bit obsessed. I was preparing for this interview, so I looked into some other interviews that you guys gave, and you had Ian Dixon on the line not too long ago, which is a great guy and a good Canadian colleague. So he's working on heart fibrosis. Don't like the way he says myofibs, but okay, well, that's between me and him. But that's just one condition where this wound healing, this initially benign process and necessary process goes wrong for reasons that we only partly understand. So there's an injury in the tissue. If this injury is too severe, so the myofibroblasts or the local environment can't really fix it in a given amount of time, or if it's chronic, then these fibroblastic cells become activated all the time. So they would still continue to produce collagen, contract the collagen, and that goes on. And they create an environment which feeds back on their own activation status. And then you end up in these conditions called fibrosis, which is excessive accumulation of fibrous tissue. And the bad thing about fibrosis is this meant to be good fibrotic tissue replaces functional tissue. In the heart example, so heart muscle is replaced by scar tissue. Same is true for lung, kidney, liver. And so you can imagine that scar tissue doesn't filter urine as a normal kidney would do. And the worse it gets, the more impeded the function of the organ is to the point of failure. And that's where a lot of the interest on myofibroblasts comes these days. I'm sad to say that it's not mostly in the wound healing. It's mostly when they go bad in fibrosis. And that's also where the interest comes from, where these cells originate from, because we need to somehow target these cells, specifically ideal. And we can only do so if we understand what they are, where they come from, what kind of markers they have, what kind of receptors they express. So we do need to understand the heterogeneity of those cells.
So what are some of the highlights of this special issue on Myofibroblasts that you don't want folks to miss?
It's almost unfair to all the authors to pick some out. So I thought about how should I answer this. Okay, so one thing is they're all great articles, and I think the heterogeneity of the articles, as you will see, or those who look into, reflects well the field, so that there are really people from very different angles looking at the myofibroblast, and they all have a very specific view. If I would like to highlight a few, or just pick three. So one is an interesting one from Chet Delast's group in Hamilton, not because they're Canadian, that's a pure coincidence here. But it's a very provocative article. They're talking again about the origin. So if you do single cell RNA-seq and look through all the data sets which are available now, you'd find collagen expressing macrophages, or cells which are classified as macrophages, which also have collagen. And we thought, or I thought, that part would be over. There was a fibroblast or myofibroblast precursor population called the fibrocyte, which was very popular about 10 years ago, which would be circulating specialized monocytes, which are thought to be precursors of myofibroblasts or fibroblasts in general. And that idea seems to be revived now with a bit of a different label. And again, we have to see where this goes. So Chet is very careful in his paper of not going overboard. But I'm the last who would say this is not possible, because if anything this field has taught me is like pretty much everything is possible, and that's why I don't have very strong opinion on most of these issues, because I've seen them coming up and being completely debunked just to be revived 5 years later. It's kind of sad, because it tells me that I'm getting old, whatever, at least scientifically. So that's one, and then there's again, a number of cool articles, but also there's one from Véronique Moulin, who's looking at secretion of microvesicles, so like a function that you wouldn't necessarily associate with the myofibroblast. Again, thinking that the cell type should contract, but there are other functions like immune regulatory functions of the myofibroblast, which we don't necessarily look at, and it's going to be cool to see how all these things interplay. So how the contractile status of the cell has an effect on its secretory activity, or collagen, or other growth factor type things. And the last certainly not the last, but from David Lagar's in Boston, who's looking at apoptosis, which is another big question in the field. It's like, okay, if you can identify them, let's imagine, how do we make them get away? And one way making them get away is just inducing their suicide. And he's working on that, and he found a number of really cool measures and specific drugs, how to specifically kill those myofibroblasts, which seem to be running around with a sort of Damocles of their head. So he calls them primed for death, and they escape death for the longest time. But if we change the right condition, they just go, well, okay, now I'm dying. So those are my personal highlights, and I'm sure everyone has their favorite field, and they'll find it in this article, in this article collection, I'm sure.
One of the things that most intrigues me is DDRs, these tyrosine kinase
Oh
receptors
yeah.
that interplay so well, and the signals can be so different, and then they can transduce signals to other cells, and that may turn off cancer or activate. Pretty exciting and interesting.
Yeah. Yeah, the DDRs, a colleague of mine, Chris McCullough, is working on them in this fibroblast context. They're pretty intriguing, right? They're collagen receptors. You would call them non-conventional, but that's probably only because we didn't think initially of them like being conventional. And you're right, they have a number of very intriguing functions. And again, I'm not sure if DDRs or DDR2 would be good markers for the myofibroblast, but if you take all those marker proteins that we know of, so DDRs, certain cadherins, certain integrins, smooth muscle actin. So if you take a collection of those, then you have a good handle on at least to see whether you're dealing with an activated fibroblastic cell or not. And you're right, the tumor field. I don't think we have something on tumor biology or cancer-associated fibroblasts in this issue, but that's the next big box of Pandora. With respect to myofibroblasts that we probably shouldn't open here.
But I'm a serial entrepreneur, and so most of my writing is always in patents and not the journal articles. So it turns out you've helped start 2 companies as well. Could you talk a little about the kind of the roles of what's the mission for the companies and what their status is as well?
Oh, I'd love to. That sounds like free publicity here. So, well, I want to claim that I have a conflict of interest here. So, yeah. So the first company, I think, yeah, that was the first company. When I was working back in Switzerland and at the Swiss Federal Institute of Technology, I was surrounded mostly by physicists, engineers, which a lot of them had no clue about biology, but they're very smart people. And what pains me to say that is you don't have to study biology to understand biology, but you probably have to study physics to understand physics. And at that time, we were toying around with ways to control the phenotype of those myofibroblasts. The thing is, if you take fibroblastic cells out of any tissue, any organism, any species, you put them in a plastic dish with 10% serum, they all become some kind of a myofibroblast. So these are not the cells in the tissue where you took them out. They're activated by just the simple fact that you took them out of their cozy environment. But we needed phenotypes which are non-activated. So at that time, the literature was full of different ways of coating substrates or coating dishes with soft little layers, which were known to suppress activation of cells. So myofibroblast are mechanoresponsive. And if you put them on a soft substrate, they would not activate. And so we use silicones, other people use hydrogels, and that worked very well for us to just do general culture on silicone-coated plastic dishes. And at the time, then we had a lot of collaborators asking for the substrates, and I had a technician who at some point said, you know what, I can't do any experiments anymore because we're sending out those substrates. All I do is making these substrates. And I had a PhD student in the lab who had an entrepreneurial spirit, said, why do we do this? Why don't we just sell these things? So that was the first company, which is called Excelnus, and they still sell those substrates. Pretty much any kind of culture substrate you can think of with the surface. And then the other company where I was not a founder, but I'm still advisor, is we found ways of micro patterning surfaces. So fibroblasts would attach but would not activate. That was a complete fundamental study. Really nice imaging. At the same time, I had a microengineer in the group who helped with making these patterns, and a plastic surgeon from the University of Geneva. And the plastic surgeon told the microengineer, you know what, we have this issue in implants and silicone implants that we place in people, they create fibrosis. And 10 to 30% of those implants have to be reoperated because of the bad fibrotic capsule that develops and deforms the implants and creates pain. And they had the idea to transfer those patterns we had onto a silicone surface rather than on a glass cover slip. And well, that's the second company. So they're more, well, Mitch, you'd be interested in that. They're more interested in developing patents and and selling patents to the silicone industry rather than making their own silicone implants.
Interesting. Really cool. So Boris, I want to thank you for joining us for this brief special issue feature of the Fireside Chat to focus on myofibroblasts. And I thought as I say, I can't even find the copy of my journal because I think it's at San Diego or San Antonio. But anyway, it was just the work in that issue was just wonderful. And thank you for being one of our editors in chief. And thank you for all the Wound Healing Society. It's tremendous.
Well, thanks both for having me. That was real fun.
We'll look forward to chatting again soon and featuring other peoples in wound repair and regeneration. Take care and have a wonderful rest of the summer.
Okay, you too. Thanks,
Cheers.
Mitch. Thanks, Kyle.